This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 49, recorded on January 24th, 2013. Hey everyone, I'm Vincent Racaniello, and we're back. Here's Dickson Despommier.
Hello, Vince.
It's been over a month.
It has.
Look, the room is empty.
Yes.
It's just you and I.
We
lost our audience.
We did.
Let me ask our audience if they're still here. Anyone here raise their hand? No hands.
No, wait, Vince, I can see lots of hands going up over there.
Where?
You just have to use your imagination.
She's a clever guy. How have you been, Dickson?
I've been very well.
Happy New Year.
Thank you. You too.
Happy holidays.
Yep, you too.
You didn't answer any of your emails. I sent you all these greetings, and you didn't.
Yes, I did. I answered some of them. Come on, I was in the wilds of New Zealand. What do you expect?
Did you have a good time?
I had a fantastic time.
So you relaxed, right?
I'm laid back as heck now. I mean,
You didn't do any work there, right?
I tried not to.
You didn't do vertical farm proselytizing or that sort of thing?
Nothing.
And where in New Zealand were you, for some of our listeners who are there?
Right. Well, we began our trip together on December 18th in Auckland, because all the airplanes internationally land in Auckland first, which is in the North Island, which is largely formed by volcanoes. And then we took a plane from Auckland to Christchurch, which is in the South Island, which is not mostly volcanic and not formed in the same way that the North Island is formed. And in fact, there are two separate islands that have just come together because of plate tectonics, which I learned while I was there. I should have known that before I left, but I'd only been to the North Island before that, for purposes of trout fishing. Okay, that's why I go there. But I told my wife that I've never been to the South Island. I know it's beautiful. Let's go. And she loves mountains. And so she wasn't disappointed. We saw lots of beautiful scenery. We went to the rainforest. It has a huge coastal rainforest, which is temperate in nature. And a portion of it is called Fjordland in the southwestern corner of the South Island. It's a place where it's impossible to build a road because it keeps washing out. They get 20 to 30 meters of water in rainfall per year.
It's good, isn't it?
That's almost a record for the world.
You'd like to have water, right?
This is true. And just over the mountains, it's desert. So that's to be expected as well. So,
how many people live in New Zealand?
A total of 4 million.
All right. How many listeners do you think we have in New Zealand?
I know we have at least one, because they got in touch with me before I left and said, when you get to Otago, which is in the southern central region of the South Island, you should contact me. And
you didn't?
No, I didn't hear back from him.
Oh. So there's at least one.
There's at least one.
If there are any others, please let us know. I'm curious.
Yeah, no, I would love to.
Actually, I could look at the stats and tell you, but it's more interesting if they actually personally just drop us an email, say, I'm in New Zealand and I listen to TWiP.
It's some of the most beautiful scenery we've ever seen in our lives.
They shot the Lord of the Rings there, right?
They shot part of it there, that's right. Not all of it, but part of it, that's true.
Some of those soaring vistas of the mountains and all that.
Yeah. No. And Fjordland was very prominent in there.
Do they have parasites in New Zealand?
Do they have parasites?
What kinds?
They used to have a lot of hydatid disease. Sheep,
that's the sheep one.
Dogs. That's right. It's a tapeworm of dogs that has the larval form in sheep and humans, and they had big problems with that for many years. And then they don't have that problem at all. But they still have Fasciola hepatica, which is the liver fluke,
right? And
it's transmitted from sheep to sheep by eating watercress. So that's a more direct life cycle. It still requires a snail, however, and it's an aquatic thing, but they still have it, and so I'm surprised because there's a pretty good drug for it, and it's more difficult to eliminate them. Much more difficult.
It's all no other parasites?
I wouldn't say that.
No malaria?
No, that's never been. It's not a tropical country, so they would never have malaria.
Is there any dengue there?
They wouldn't have that either, no.
Yellow fever?
Nope. Those are all tropical.
Yeah, I know.
I know. They have no tropical disease. It's not a tropical place.
Okay. No reduvid bugs?
No reduvid bugs. They've probably got some form of Kinjin, like West Nile, like some flaviviruses might be
they
there.
They must have toxo, no?
Oh, they've got toxo. Sure, they do.
And that's one of the papers we're going to talk about today.
Yeah, that's right. They've got toxo. They probably have cryptosporidium as well, because they have a lot of cattle. And they're big products. New Zealand has a very interesting set of industries, and one of them is sheep, of course. Everybody knows about the sheep. But there's a lot of wheat that's grown in the southeastern portion of New Zealand, which I was surprised to see. But I circumnavigated the entire periphery of the southern island looking for a place to trout fish, because they had a huge storm that came through just before my wife left. And every river in the South Island was, as those fishermen would say, it was blown out. That it was so much rain that the rivers exceeded the banks and was largely unfishable for at least a week everywhere around that whole country. The entire country was like that. Amazing. So it shows you what the environment can do for a situation.
Well, you're back for a few days.
That's true. Then I'm off to Sweden, of all places.
When do you go to Sweden?
Next Monday for a conference on urban agriculture.
That's probably a few days, right?
A few days.
Then you're back, and then what's the next trip?
I go to Germany.
In February?
March.
So you're here for February.
Oh, yeah.
Are you sure we could do a couple of TWiPs?
No, we could do more than a couple. We could do one a week if you'd like.
Yeah. Okay.
Sure,
Are there any parasites that we should cover?
Yeah, there's tons of them. And tons. Tons.
Okay. Well, I saw you for the first time on Monday, right?
That's right.
No. You weren't here on Monday, were you?
I was here Tuesday.
Tuesday.
Yeah, Monday was Martin Luther King Day. Nobody came in Monday.
I came in on Monday.
Well, some people came in.
I had work to do.
Right. No, I got back Sunday, so I figured...
And my kids had school because they...
Did they really?
Yeah,
because of Sandy.
They
That's
had to
right.
make up for that 10 days that they missed back then.
Wow.
Well, in your absence, I picked a couple of papers.
Right.
Because I figured it was the minute you stepped in the door, say, here, read these, and we'll talk about them.
Sure.
I thought they were interesting.
They were both interesting.
You can tell me if they weren't.
No. I thought they were.
Because this is your show, and usually you pick the subject. But
usually.
Since you weren't here, and I wanted to get a running start,
you did a good job.
Back,
no, I thought you did a good job.
The first one is authored by a number of groups, people from different groups, but one of them is right here in our department. My chairman, Shankar Ghosh. But there are also groups from Brazil and MIT and Mexico City. There's this group in Brazil, the Federal University of Minas Gerais. And it's interesting, Minas Gerais. You remember in The Lord of the Rings, Minas Tirith?
I'm not a big Lord of the Rings fan.
Minas Tirith was the name of one of the city. Minas. What does Minas mean? Well, I should just look it up, right?
I think it might mean minor, but I'm not sure.
You think?
I don't know.
Minas. It refers to places. Oh, it's just a Wikipedia thing. May refer to, Minas Tirith, a fictional city in Canada, in J.R.R. Tolkien. Minas Morgul is a fictional fortified city. Minas Gerais in Brazil. Minas Uruguay. Minas Department in Argentina. So it can be used. What does Minas mean? That's what I want to know. I know this has nothing to do with...
I know one of our listeners out there has that answer. We'll probably get a whole bunch of them.
Let them tell us, because I'm not going to look it up.
Right.
I was always struck by that similarity. Anyway, we're going to talk about a few papers. We do have a bunch of emails that have accumulated in your absence, which we will address.
Well, good.
And one of them has to do with your breathing.
Tell them I have to breathe no matter what.
Well, they didn't say not to breathe.
I know I'm a heavy breather.
First, it's okay. No problem. The first one has to do with Toxoplasma gondii.
Yes.
The title is Combined Action of Nucleic Acid Sensing Toll-like Receptors and TLR1112 Heterodimers imparts resistance to T. gondii in mice.
Right.
So, T. gondii, we talked about many episodes, right?
Yes.
It says here that this toxo infects 300 mammalian and 30 avian species.
Correct.
And they say felines are the definitive hosts, and mice carry the
mostly
parasite.
rodents. Mostly rodents.
The natural intermediate hosts and main natural reservoir, they say, are mice.
Yep.
Would you agree with that?
I would.
They call it a coccidian parasite.
It is a coccidian parasite.
I just want to check that they wrote this correctly.
It's an apicomplexa.
Yeah, it is an apicomplexa. It has this organelle called an apicomplexa, right?
That's right.
Which came from a plastid from plants.
Yes. Weird, huh?
Well, our mitochondria came from bacteria. It's not so weird. These things happen.
These things happen.
They say 1/3 of the world's population carries a chronic and asymptomatic infection with T. gondii.
That's probably the most popular infection throughout.
Yeah, you've said it was the most successful parasite. I think that
is.
was the name of
Yeah, that's
the episode.
Right. That's right, and it is.
And most of these people are fine.
Correct. And except when they start to get old.
So that's interesting because it says here that in immune-compromised individuals, the dormant parasite becomes virulent, leading to reactivation and chronic infection.
That's correct.
So I guess as you age, you get immunocompromised, right?
Some do, some don't.
You seem to be pretty healthy.
I feel good.
You don't get sick.
For the most part, that's true.
Yeah, I don't see you with infections very often.
I've had a cold.
Yeah.
Every now and then.
Well, you don't have toxo either.
I don't know that. How do I know? I've never been tested.
You haven't been?
Nope. Why would I have been tested?
I don't know. Curiosity.
Well, that's true. I could have been curious
Curiosity
enough.
killed the cat because they ate the mouse, right?
Satisfaction brought them back.
So they discuss a little bit the basis of immunity. This paper's all about immunity because that's what they're exploring.
Correct.
And they say that the main correlates of immunity for
Yes.
toxo. You have to correct me if I'm wrong.
I can't correct you if you're wrong because I might not know.
All right. Well, that's fine. You could try.
I can tell you what I do know.
It's immunity or resistance is dependent on T cell immunity.
Yes.
Particularly T cells that make interferon gamma,
Correct.
and also T cells that lyse the parasites, either the so-called
That's right.
CD8 effector lymphocytes, cytotoxic T cells is another way, right?
Yeah.
So these are important.
Very important because without them, you wouldn't be able to stimulate the toll-like receptors.
You know how they would come to such conclusions that these kinds of cells are important?
Well, because the animals eventually do develop immunity. And so how else can you do that unless
Well, you
you
could take them away from mice. You can deplete specific subsets in mice and then infect them.
Remember, mice and humans are totally
Yeah,
different.
they're not the same, right? So you do studies in mice, and then you say this is what happens in mice.
Right.
And in this paper,
It's very obvious there's a difference.
All this paper is done in mice. Yes, and there's
I would have written that title differently, of Mice and Men.
Yeah, I think the title isn't terribly good.
Of Mice and Men.
Everybody gets to write their own titles.
That's true. But that title's been used already, Vince, I'm afraid.
Mice and Men?
Of Mice and Men has been already used.
I think we even used it.
We have.
So you can deplete specific parts of the immune system in mice, and then
absolutely.
ask. Let me ask you: when you infect mice with toxo,
Yes.
you do it IP. Or you can do it
You could.
anyway, I guess.
It doesn't matter.
And what happens?
Well, if you infect IP,
Yeah.
then the tachyzoites are taken up by the exudate that develops inside the peritoneal cavity, and
Right.
the macrophages take them up. And we know for a fact that this organism has the ability to inhibit the fusion of lysosomes to the parasitophagous vacuole, so the parasite survives, and then is transported from the peritoneal cavity to the bloodstream, and it's distributed throughout the body. And then when the macrophages die from their initial infection and they
Right.
release tachyzoites, they then get taken up by more cells. And this parasite has the ability to infect every single cell.
Do the mice die?
Some do. Depends on the strain
It's probably also the dose, right?
and the dose. Remember when John Boothroyd was here, he talked about strain differences
Yeah.
and things like this. But eventually they all will
Yeah.
either succumb to the infection or harbor a latent infection, like we come to do it.
Okay.
If they died from it, this would be a very poor parasite, wouldn't it? Because then the mice
Right.
wouldn't be able to transmit it to the cats. Cats don't eat dead mice. They like to chase
Right.
their food.
All right. So T cells that make interferon gamma
Right.
and cytotoxic T cells are important. They also say what's also important
Yeah.
is a protein called MyD88.
Right.
Oh my D88. Now,
Right.
MyD88, because you can delete the gene for this protein in mice, and you can find that all of a sudden they become killed
That's right.
universally by
That's
toxo.
right. That's right.
MyD88 is an adapter. Let me show you this picture here.
Please do.
I have a picture of toll-like receptors here.
Okay.
Toll-like receptors
Yes.
are sensor molecules on cells. They can be on the plasma membrane, or they can be in the inside of endosomes.
Right.
Because a lot of microbes get taken up by that pathway. They sense when something is foreign.
They do. This is part of the innate immune system. Is that what you're trying to tell me?
It is.
Great. Because we've had this discussion before.
Have we?
Also. We have, only not in TWiP.
In TWiP?
We had it in TWiP.
So the sensors, and there are other sensors besides toll-like receptors or TLRs, their job is to sense something foreign. All right. And they do so by recognizing molecular patterns that are foreign. It could be nucleic acid. It could be peptidoglycan, glycoproteins. And did I say nucleic acids?
You did.
Yes, I did.
Both RNA and DNA?
Yeah.
Because that includes all the viruses.
Yeah, even
then?
single versus double-stranded RNA. Too.
Sure.
So the viruses typically get sensed.
Or the bacterial flagellum.
Yeah, exactly. So flagellum is another one, right? And there are over a dozen TLRs, and
it's amazing.
What happens is, if, let's say, there's a TLR on the cell surface, when it recognizes a ligand, it starts a signaling pathway.
Right.
That eventually leads to the production.
Is that phosphorylation dependent?
Phosphorylation, yeah.
Mm-hmm.
And there's a molecule in that pathway which is needed for all toll-like receptors except one. It's called MyD88.
Huh.
So if you knock out MyD88, you're basically messing up the toll-like receptor sensing ability.
What does MyD stand for, Vince?
What does MyD88 stand for?
Yeah, it's an abbreviation for something, no doubt.
You always ask me abbreviations.
Well, to bring me up to date, to bring maybe the parasitologists are not as familiar with that molecule because we don't involve it mostly in our research.
Do you really need to know what it stands for?
I would be pleased to learn what MyD stands for. You think I'm a heavy breather? You listen to Vince right now.
I got it.
He does.
Myeloid differentiation primary response gene number 88.
Goodness gracious.
Yeah, you wish
No
you
wonder they abbreviated it.
So it's an adapter, which means that
got it.
For the signaling through the toll-like receptor, this protein is essential.
Okay.
And that signaling eventually turns on the production of interferons and cytokines in the nucleus, so that's the antimicrobial response. Right. So if you knock out MyD88, you really impinge on all the TLRs. So what they did is they knocked out MyD88 in mice and then put toxo in them.
How did they keep them alive long enough to do that?
Oh, you don't need MyD88 to just live. You
really?
keep them in a clean facility so they don't get infected, right?
Wait a minute. I thought we were always being infected.
In a clean facility?
Doesn't matter. Bacteria everywhere.
Yeah, they're not notobiotic.
They're not notobiotic. So
say that 10 times fast. Not notobiotic.
I used to have to do that at Notre Dame.
I used to work on notobiotic mice.
That's right. You do.
So there are facilities where you can raise your mice for research.
Yes.
They're called barrier facilities.
Yeah, I've done
So you don't have viruses, exogenous viruses and bacteria. I know, it all depends how careful you are, because when you walk in, actually you have to wear a mask and gloves and head cover and a lab coat and everything.
Okay.
Some are better than others.
I'll suspend belief for the moment. But
you can raise MyD88 knockout mice quite easily.
Okay.
Of course, if you put them in a non-barrier, they might get infected and die of infections. Okay.
But in this case, we're going to purposely infect them with toxo, and
well,
then
when they put toxo in, they die because they can't mount a response. So we think, or the authors and people who have done this work think that TLRs, toll-like receptors, are important for defense against toxo. All right.
Absolutely.
So that's one of the experiments.
Yep.
And what they say here is quite interesting. If you knock out MyD88, you don't get IL-10, IL-12, TNF alpha, and interferon gamma produced. And they say all of these are important for surviving a T. gondii infection.
Right.
Now, one of those, IL-10,
yes,
is actually a regulatory cytokine. It prevents an excessive inflammatory reaction.
It's the yang of yin.
Yeah. So when you make cytokines, it causes inflammation. It brings in lymphoid and macrophage cells.
You have to downregulate afterwards, right?
To try and clear the infection. That's called inflammation.
Right, sure,
TNF
sure,
causes fever.
Yep.
systemic symptoms, malaise,
Yeah.
muscle ache. Those are all caused by cytokines.
In fact, there's a special issue of Science that's devoted to inflammation that I just received in the mail, and it was quite informative.
And you have to regulate it.
And you have to regulate it.
Because if you have too many, you get a cytokine storm and you can die.
What are diseases caused by overinflammation?
Well, do you remember the toxic shock syndrome
Sure do.
episode of years ago? You know those tampons?
Yeah.
So many women were getting infections and
Correct.
getting toxic shock because of the production of too many cytokines.
Right.
That's the first time I came into my radar, I think.
Uh-huh.
It took a long time to sort that out. It's not
Yep.
just a regular old bacterial infection, but it's one that induces a cytokine storm. Am I talking too much?
Not at all. You're talking just enough.
So IL-10 is important for dampening this response. All right.
Right.
So you knock out MyD88, you die of toxo because you don't make these important cytokines.
Yep.
Okay.
Yep.
Now, what is recognized by TLRs on the toxo? That's the real question, right?
Yes, because they don't contain flagellum, even though
They don't have flagellum.
they don't have a flagellum.
But they do. They don't. They have nucleic acids, right?
They do.
And presumably they have other proteins on their surface.
But the only way to get them out of the cell and onto the TLRs is to lyse open the organism.
Well, remember, some
And that's why the second mechanism is important.
TLRs are on the inside of the lysosome.
Yeah, but they prevent fusion of lysosomes. The living organisms prevent the fusion of that, so that's not going to work. It's not going to work.
We'll
Vince.
keep that in mind. Hold that in your pocket because we're going to come back to that.
Hold that in my Parasitophorus vacuole.
So recently it has been shown that a profilin-like protein activates TLR11.
I looked
What's
up.
profilin?
It's a molecule which interacts with actin.
Is it related to profane?
No, only if you mispronounce it.
Where is it located in the T. gondii?
That's a good question, actually.
Is it on the surface?
It's got to be on the
Yeah,
surface.
I would guess so.
Because otherwise it couldn't interact with the actin filaments in the forming vesicle that it induces with its apicomplex secretions.
An actin binding protein found
Yes.
in all eukaryotic organisms.
Correct. It's a rather interesting molecule, I thought. And it's important for gliding. Okay, this organism, since it has no overt locomotor molecule,
Yeah.
or it doesn't exhibit amoeboid motion, but it still moves. If you look at it under the microscope, it will move. It's called gliding. And profilin, I presume, is the molecule that enables it to do that.
So the finding that profilin is recognized by TLR11
Yes.
caused some investigators to knock out TLR11 from
Right.
mice.
Right.
But they don't get very sick.
Nope.
You don't get very
That
sick, so
was
that doesn't explain the
interesting.
whole thing.
That was interesting, isn't it?
And so then what they did was make another gene knockout of a different protein, which we need to discuss a little bit. And
So
this
wait a
is-
minute. Let's back up a minute.
Yeah,
and
we
say
can back
why don't they get very sick from just knocking out the protein on the host that would prevent the organism from entering the cell? If you knock it out, then profilin can't interact with it. Correct?
Well, profilin is interacting with TLR11 to give an innate immune response.
Right.
So if you knock out TLR11, if it's the only thing that's important for sensing toxo,
And it's on the surface, right?
which TLR11?
Yeah.
Yeah, TLR11 is a surface guy.
Right. So that probably has something to do with the entry of the
Maybe.
But once it gets inside, then all the rest can still be active.
All right, so apparently it still gets in when TLR11 is there, and there must be other things sensing toxo. That's the point. If you take TLR11 away, the mice can still mount a response.
Yes,
That's the
indeed.
key. Okay?
Indeed.
So there's another protein that we have to introduce. It's called UNC93B1. You want to know what UNC stands for, right?
Everybody does. Come on. It's not just me. I'm the voice of our listeners saying, why are you throwing all these terms at me?
There's actually a Wikipedia page for UNC.
Oh, really?
Yeah. UNC is...
Is it a good page or a bad page?
I don't. Here now, it doesn't tell us what.
The UNC
It's
says.
actually. It was originally discovered in C. elegans, actually,
Oh, no kidding.
but I don't know what UNC stands for. I'm sure it's a wonderful name,
Me too. But not very descriptive in terms of what it actually
and it would just
does.
take me too long to find it out. Anyway, UNC 93B1 is a chaperone for toll-like receptors.
Chaperone.
So, toll-like receptors being membrane-bound proteins, they're produced in the endoplasmic reticulum.
Yes.
And then they have to be shuttled to either the plasma membrane or the endosome, depending on which one.
Right.
And
go on.
The chaperone is important for that.
So it guides it to the proper cellular location for insertion.
Exactly. So if you take away UNC93B1, you mess up the toll-like receptors going to the right place, and so you basically also interfere with sensing. Okay?
Right.
A little bit of an aside here. UNC93B1, people who have amino acid changes in that protein are predisposed to getting lethal herpes encephalitis.
Really?
What does that tell you?
Very important molecule.
For sensing herpes infections.
And that's a very common infection.
Yeah, so everybody has herpes, right? But not everyone.
Possibly.
Herpes encephalitis is rather rare.
Yeah.
And of the rare infection, some people die of it, and those people apparently have a mutation.
Wow.
Some of them have a mutation in the
Wow.
gene encoding UNC93B1. So it's very interesting.
Yep.
And as we will see, this is also important for toxo.
Fascinating.
So you take mice, you knock out UNC93B1,
Mm-hmm.
and they get very angry. They get angry, right? I mean, what's the result? I'm going to have to edit this now. All right. So UNC93B1 deletion mice are very susceptible to toxo infection.
Okay.
All right. So,
proving once again that toll-like receptors on the surface are important for recognition and immune response.
Right. Now,
Yes.
UNC93B1 was always thought to be most important for the chaperoning of nucleic acid sensing toll-like receptors, which in mice are 3, 7, and 9.
3, 7, and 9. Isn't that an interesting selection? How many
Those are the
toll-like
ones that
receptors are there?
Well, I said there are over a dozen.
Right.
Human and mice. They have different numbers in humans and in mice. So, if you knock out...
Wait. Are there UNC molecule-like things that chaperone the others to
I'm
their
not aware
proper
of them.
place?
I'm not aware of them. There may be, but I'm not aware. I'm just aware of UNC93B1.
Right.
Even though I don't know what it stands for, I'm aware of it.
Fine.
All right, you knock out UNC93B1, the mice are susceptible to toxo. If you do a triple knockout of toll-like receptor genes 3, 7, and 9, they're not as susceptible to infection.
Right.
So that tells you that there must be something else involved, right?
Correct.
And that was the whole point of this paper.
That's right.
They wanted to figure out what was going on.
Yeah.
And the bottom line is, first of all, they show that TLRs 7 and 9 do sense toxonucleic acids. Okay? TLR7 senses RNA from toxo, and TLR9 senses DNA. You do this in macrophages that are lacking one or the other toll-like receptor, and then you add nucleic acids. Okay. They also found that TLR11 and 12 recognize something else, and that is this profilin of toxo.
Right, which it uses to gain entrance into the host.
Does it?
That's what it suggests. It interacts with actin in a gliding mechanism. So it's
That's
probably
what toxo uses to get in?
Yeah, it's probably important for it to make this parasitophagous
Yeah, but I don't think
factor.
TLR11 is important for that, for entry of the toxo, because you can take it away and they will still infect. It's for sensing, remember, it's just for sensing. So
I wasn't talking about TLR11. I was talking about profilin.
So profilin is a toxo molecule.
It's a toxo molecule.
So you think it's binding something on the host cell? Okay, well, that may be.
Yeah.
Do the experiment.
Well, they've already done it in other cells and shown that the profilins in those cells bind
Okay.
with actin.
I'll take your word for it.
No, really. I looked it up.
Okay.
I actually read something on this.
So basically, they find that TLR11 and 12 recognize profilin. So that's what was missing, because if they now make a 3, 7, 9, 11, 12 knockout, quadruple, yeah, now they're as susceptible as the UNC
That's incredible.
92. What was it? B3.
See, that's what's wrong with that.
I know. I can't
Not knowing
remember
what
it.
they mean.
I remember my D88 because I.
Yeah, that's right.
I actually think about it a lot.
Yeah, right.
But UNC 93B1, I don't.
I don't spend a lot of
So
time.
apparently 93B1. So TLR11 and 12 are not nucleic acid sensing toll-like receptors. They're
No.
recognizing profilin, right, which is a protein.
Which, if you think about it, another one recognizes flagellin.
Yeah,
so that's
for
an
bacteria.
external protein as well. And so this is a very interesting set of molecules here.
Very interesting. So basically, ANK93B is controlling TLR11 and 12 in addition to 3, 7, and 9. So it's not just working to chaperone the nucleic acid sensing.
Yeah, it suggests another thing here, Vince, too, and that is that the relationship between Toxoplasma and intracellular parasitism has been around a long, long time.
Why is that?
Well, because there's a specific TLR receptor for just that particular protein that's present only in toxo. I don't know of any other intracellular parasite that uses it to get into a host cell. I don't think malaria uses it. Crypto probably doesn't, but we don't know that because it's an apicomplex. It might use it, and so is malaria, but I think malaria doesn't use that as a mechanism. It uses glycans, like blood group substances.
Okay.
So here's a molecule specific just for toxo that all mammals, maybe even all animals have because it's such a promiscuous parasite.
By the way, TLR11 and 12 are endosomal.
Really?
So what they think is happening is that in mice, the role of this is to recognize toxo in the endosome. Now, I know you said before that they prevent this from happening, but I don't know how that's compatible.
No, I didn't say that at all. I didn't say that.
So when toxo is taken into a cell, it goes into a parasitophorous vacuole.
It creates the parasite by secreting these substances out of the conoid, the region in the front of the parasite. Remember? Right. John Boothroyd.
And so that would normally fuse with a lysosome or an endosome, right?
It prevents the lysosomes from fusing.
I know, but somehow these are being sensed in the endosome.
Right.
So how does that happen if you say they're in a parasitophorous
Well, that's
vacuole?
maybe they don't answer that question in this paper.
So they say here the protozoan ligands that are recognized by the toll-like receptors appear to reside inside the parasite, DNA, RNA, and profilin. So profilin is inside the parasite, all of which are released from the parasite when it is killed in the phagolysosome.
Yeah, well, it's killed, but what's killing it?
The phagolysosome. The enzymes and the
what kills the
low
parasite?
pH. I don't know. You tell me.
But remember, this parasite can live in a macrophage and kill it.
So why is this being killed in an endosome? Tell me.
Well, John Boothroyd was here. He could speak to the issues better than I can. But in the beginning of this infection, there's no immunity. And this parasite has its way with us, and it can infect almost every cell.
Uh-huh.
So what you're looking at here is the development of immunity, which is IL-12 dependent and interferon gamma mediated. That's the immune mechanism that prevents infection in other cells after this thing develops an immune response.
Right.
But why would some of these original tachyzoites die when there's no immunity developed yet? And the answer is that maybe they all don't live an equal life or something.
Maybe some of them are sensed. Some of them, I don't know the answer. Maybe someone knows. But the point is here that some of the toxo make it to the phagolysosome, and there they're destroyed, and their ligands, DNA, RNA, and profilin, are then engaged by the toll-like receptors, and that activates the production of these mediators.
I got it, but
are important for resisting infection.
But that doesn't happen in the beginning of the infection. It only happens in the middle or the late stages. No, I'm telling you.
Yeah, I agree. I think you have to think of this not as an all or none, but as a stage.
So, yeah, what stage of the infection are they describing here? That's what I was asking.
Well, they don't actually say.
Because you can take a lawn of fibroblasts and kill it with toxoplasmosis.
Sure.
You can just throw them in there. There's nothing stopping this parasite from killing every cell.
Yeah, they don't actually address the parasitophorous issue.
Right, and that's important because really the way you advertise this parasite to students, particularly medical students, and that's what I taught for many years, as you know, is that this is the world's most successful parasite. So what makes it successful? And the thing that makes it successful is two things. One is that it can infect every cell. Two, you catch it by eating meat, usually, raw meat or undercooked meat. And three is that it's very slow to develop immunity. But once the immunity develops, that wherever you have Toxoplasma in cells starting to reproduce, their reproductive cycle is interrupted, and they produce something else instead. They produce these pseudocysts that are loaded with the bradysooites. Remember? That's a resting stage. It's more or less like the same equivalent to keeping tuberculosis intact by creating these pseudotubercles in your lungs, but it can reactivate if the immunity wanes. The same is true for these cysts. So the real question I have is, when does this immune mechanism that's being described here come into play? It certainly doesn't come into play early on in the infection. It can't, because this parasite really has its way with us for quite a while, and you can get very sick from this, by the way. You can develop swollen lymph nodes. You can develop mono-like syndrome. You can be sick for months with this, and with fever and all kinds of things. And people with immunodeficiencies have even a longer-term infection with this. So it's quite interesting to see what's going on.
I don't know, because, of course, these are done, a lot of these experiments are done in macrophages.
Right.
So the timing is not clear with respect to a mouse infection.
Correct.
My understanding is that in a mouse where infection doesn't kill it, there has to be some regulation of multiplication of the toxo. Maybe this is how it happens. It doesn't eliminate it, but it keeps it in check because they remain infected. They're chronically infected, right?
Right. I mean, I think that not all the tachyzoites survive when they're released from an infected cell. Some of them may die and then release their RNA and DNA molecules and their profilins as well.
Profilins.
Yes.
Or profilins, whatever.
Profilins.
Don't defile. Right. Let me tell you something else which is really interesting. This is the last thing we'll talk about for this paper.
Please.
TLR humans don't make TLR 11 and 12.
Right.
So 11 is there.
It's a pseudogene.
It's a pseudogene, so
right?
we don't make the protein.
That's
the 12
right.
gene is not even there.
Exactly.
So they say, they speculate, because mice are the natural intermediate host, they speculate that 11 has been maintained in mice by selection over the years, right?
Right.
So you put pressure to keep 11, because if you don't have 11, you're going to die and you're going to be out of the pool, right?
This is true.
You're out of the pool.
That's right.
So in people, what's regulating toxo
Good
infection, right?
question.
So they speculate that 7, 8, and 9 are important for humans, just like, 7, 8, and 9 are nucleic acid sensing toll-like receptors in people. And we know in mice that 3, 7, and 9 are sensing DNA and RNA of toxo. So they're speculating that in people, these must be important for sensing, but maybe there's something else in people that is sensing profilin, for example.
Sure.
They also say it remains unclear. Look, so although it is considered an accidental intermediate host, people,
Yes.
1/3 of the population is chronically infected.
This is true.
It remains unclear why it was downgraded to a TLR11 was downgraded to a non-coding gene in humans with all this pressure on humans by toxo. That's the point. So at some point we had 11 gene and it became a pseudogene, right?
Yes.
Because it didn't enter us as a pseudogene. There's no selection for that. We had to have TLR11 gene at one point, and then it became a pseudogene because we didn't need it. And what they're saying is, how could it be that we didn't need it? We got all this toxo.
Yeah, wait a sec here. Why is it the most successful parasite in the world, though?
It just spends a lot of money to get its place.
It doesn't kill its host.
Right.
No matter what the host.
So you're saying it's not putting pressure on us?
Right.
Does it kill mice?
No.
Yeah, but they have 11.
But they don't kill them.
But why do they keep 11? Maybe there's something else.
No, that's to protect the mouse. The mouse...
No, Dickson, the mice are not killed by toxo.
I accept
and
that.
humans are not killed.
And they both develop pseudocysts in their brains.
Yeah, but mice have 11 and humans don't.
Right.
So what I'm saying is maybe the pressure to keep 11 in mice was from another microbe.
Right. It had nothing to do with toxo.
Maybe.
I would
There's
agree
no
with
evidence here that 11 was maintained by toxo.
I agree with you.
Okay, good.
I totally. I agree with you.
Right. So they say there must be another toll-like receptor in people that's sensing profilin or something.
This is one of the most gentle infectious agents in the world.
The gentle giant?
It is. It doesn't want to hurt anybody. The only people that get hurt here are immunodeficient people or people with AIDS. And the reason for AIDS and toxo linkage, although John Boothroyd was he was ambivalent with regards to his opinion about that, and I accept his ambivalence as proof that it's not altogether proven. But when AIDS patients reach a certain level of depression of their CD4 and CD8 cells,
Yeah.
when they go below a certain level, the IL-12 levels drop. And when the IL-12 levels drop, their latent Toxoplasma infections reactivate.
Oh, interesting, because that's, as we have seen, is important for controlling toxo. Now we finally understand something that you said way in the beginning TWiP. Actually, you said that way in the beginning of TWiP,
I did.
Did this light bulb just go on in your head?
No, it went on in your head.
It's always been on in yours?
It's always been on in my head.
Hey, wasting electricity.
No, I use solar panels. I don't have any hair on the top of my head.
I was going to say, the head is very
I've got a
good
solar
for that.
panel for this. So that's the connection, and that's the reason.
Yeah.
That only came out.
Is very important.
It only came out during the AIDS epidemic.
So people have exacerbations of toxo. Yeah.
Well, they used to. They don't anymore because they take the
yeah, of course. But there are all these opportunistic, so-called candida.
Yeah,
Pneumocystis, right?
Sure.
Coccidio, I think, also.
Sure, strongyloides. Even it turns out that malaria is part of that same scene.
Wow.
But in a more subtle way. Yeah.
Well, like any good paper, this actually raises more questions than
right.
Some guy's making fun of my blog post saying, "Oh my, headline writers are doing mistakes." How about that?
Oh, gee. Thank you for informing us.
That was an aside that no one will understand unless you go to virology.ws.
And they probably will.
All right.
They probably do already.
Let's
move on.
Talk about the next paper, which I thought was cool because it's a review in PLOS Pathogens. It's called Make It, Take It, or Leave It, Heme Metabolism of Parasites.
Right.
It's from Czechoslovakia.
Indeed.
And these guys' names have all these accents over them that are like an upside down.
Yep.
What do you call the French one that is like a peak?
What do I call it?
The accent.
It's an accent.
No, but there are all kinds of circumflexes and grave and
a tilde and all those
tildes,
things.
But this one is.
I don't know what this is. I don't know what the name for it is, but I know that in French they have.
It's an upside down carrot.
That's correct.
Okay.
Yes.
And we should have our listeners, that we have some French listeners, tell us what this is, or Czech listeners. Anyway, they called it heme. Heme is a tetrapyrrole. Did you know that? What is a tetrapyrrole? It sounds like a four-member.
It's got 4 pyrroles.
What's a pyrrole?
I also thought it was a porphyrin, and it is, of course.
Yeah. What is a porphyrin?
A porphyrin is a heterocyclic compound made up of 4 components.
Tetrapyrroles, right?
And 4 heterocyclic 6-member carbon rings.
So I know
5 members.
I know
5 members
because it's in hemoglobin.
But of course,
right.
when I took my first biochemistry course here, Vince, a long time ago.
Yeah.
I took a course with Dr. Shemin. Do you remember that name at all?
How do you say?
Shemin.
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 Oh, Shemin.
Shemin. He was in the biochemistry department, and he taught it on the downtown campus. And I took that course while I was here as a technician in 1962. And he made us, the whole class, remember the structure for protoporphyrin 9. Is
that a precursor to porphyrin?
It is,
yeah.
because Shemin was instrumental in using acetate, which was radiolabeled, to trace the synthesis of that molecule in hemoglobin synthesis. And he did it out in Brookhaven, because in those days, in 1962, it was the only place you could go to find radioactivity.
You know, I now realize why his name is Shemin.
Please.
Chemin de fer. Do you know what that means?
No, but you're going to tell me. I know you will.
It's a railroad car.
Oh.
Carriage of iron. Fer is iron.
Ah.
And iron, of course, is part of heme.
Oh. Do you know what the Spanish equivalent to that is?
No.
Ferrocarril.
Ah, nice.
Yeah,
how do you know Spanish? I don't. Okay. I'm here every night.
Yes, that's right. Try
the veal.
Try the veal.
Anyway, the heme.
So heme, I'm familiar with the heme group, of course, and so are you.
I'm not familiar. You're going to be the
found all over
source
the place
of information. I'm going to ask you questions.
Oh, and I'll see if I can get them right.
He says here, these authors say that heme and other tetrapyrroles, so there are apparently things related to heme. Right?
There is. There's cobalamin.
Chlorophyll.
Right? Chlorophyll. Vitamin B12.
Vitamin B12.
That's cobalamin.
And other things, right?
Yeah. It's a common molecule that's been used. It's a metalloporphyrin.
They say it's highly conserved throughout the tree of life.
It is.
So chlorophyll is a green.
That's right.
Vitamin B12 is involved in DNA synthesis.
That's right.
And then heme. They call this the colors of life, the tetrapyrroles. Because heme
Remarkable.
is red. And
by the way, worms
chlorophyll's green.
have this molecule as well.
Are you defending worms?
I am, because they don't appear much in this text.
All right.
Go on.
What's the best known function of heme?
In human beings with hemoglobin to carry oxygen
That's right.
and to deliver CO2 to the lungs.
It says here, unicellular organisms do not need to transport oxygen, but may still use globins or other hemoproteins to sense diatomic gases and to avoid oxidative or nitrosative stress. Do you understand that?
I do.
Please explain.
Well, what they're doing is using heme as a scavenger molecule to keep away the harmful oxidative molecules that might have something to do with oxidizing their proteins or making their DNA a little
So
bit strange.
diatomic gases, so oxygen,
Like O2,
nitrogen,
N2,
anything else?
H2.
So these would be bad for the parasite.
For some.
For some, and so they use heme-like compounds
trap.
to trap them. Okay.
Yeah.
I see. So these things are important. They call them reactive oxygen
Very
species, ROSs. And so these, and then they also say many parasites have adapted to anaerobic environments, so they don't need heme to carry around oxygen, but they still do have heme proteins.
Because they need to protect themselves from oxygen.
Right. Okay.
And then that's where the worms come in. That's when I wanted to add the worms are like that too.
The whole focus, or one of the major foci of this article, is using heme metabolism as a target for new anti-parasitic drugs.
Right,
Okay,
That's right.
so we'll come back to that a bit later.
Yep.
All right. So they have a little introduction. Then they talk about how is heme synthesized and how do parasites obtain
Right.
it.
Did we read the title of this paper yet?
Yeah, we did. Make it, take it, or leave
yeah,
it.
okay,
Heme
fine.
metabolism.
Okay,
so you
fine.
can either make it,
Right.
You can either take it,
Yes.
or if you leave it, means you don't need it, right?
Correct.
So some don't need it. So I think we should go through those.
And for some, it's even a toxin.
Heme.
Yes.
Really?
Or a version of heme called hemozoan. We'll get to
No,
that.
we're going to get to hemozoa.
We will get to that.
So it says here, most organisms are able to synthesize their own heme, and they talk about the pathway.
Yep.
And the fact that
And that pathway, by the way, was established by
who?
David Shemin.
Oh, he did. He did.
Yeah,
really.
I told you. He went out to Brookhaven and worked with radiolabeled
told me
acetate.
that?
I did
I'm
earlier.
sorry, I just don't listen to you.
Earlier, I told you I took a course with him, and he became famous for having synthesized heme, or protoporphyrin 9, which is a precursor for heme,
Okay.
and described all the molecules that are needed to make it.
Apparently, the mitochondria and the chloroplast
Yes.
in plants are important for heme. So it's a
Very.
combination of pathways that occur there and also in the cytoplasm. I didn't realize that. Did you know that?
I did know that, actually.
So the idea is that we acquired heme synthesizing ability from this symbiont that used to be a bacteria, right, which came into us. And so the idea is we had the ability for a while, then it came into us, and we discarded most of our machinery, and now we use this mitochondrion or chloroplast. Plastid. What's a plastid?
An organelle-like laminate membrane.
Is that a
organelle.
chloroplast?
That is.
All right. Just another name for a chloroplast. One is part of the other.
It's an intracellular particle.
So a chloroplast is a kind of plastid.
Yes.
Are you sure? Because our listeners are sharp.
I'm pretty sure of that definition,
All right.
but you can, look up in Wikipedia. Forget about Wikipedia.
Oh, I don't look it up on Wikipedia. So I have a nice table here showing all the parasites and what they do.
Well,
for
not all the
making them.
parasites. That's the point.
Not all of them. They
A
have
lot of
aerobes and microaerophils or anaerobes here.
Right.
Let's just name some of these aerobes.
Sure.
That I know Leishmania, T.
Yep.
brucei, T.
Yep.
cruzi,
Yep.
Plasmodium, Toxo.
Yes.
Then the anaerobes or microaerophils, Cryptosporidium, Trichomonas, Giardia,
Trichomonas.
Entamoeba,
Entamoeba.
Nematocystis, Blastocystis. There are actually a few here that I don't recognize.
I know nematocystis and blastocystis.
In the first one, the aerobes, there was Crithidia.
Crithidia. It's a plant equivalent to the trypanosomes.
Okay. Phytomonas.
It's a free-living flagellated, like Euglena. It's a little bit
Okay,
like
so
Euglena.
that's another aerobe. The reason I'm slow at pronouncing these is because they're at a 90-degree angle on the
I
page
know
here.
that. I
And
couldn't
I
get
think
that
that's
to
really
turn
bad.
around
It should
either.
at least be at a...
No, it should be a flip dial
Yeah.
that you can actually reduce the size and put it all.
Encephalitozoan.
Yes.
What is that? That's an anaerobe.
It's a microsporidium. It's got an unusual classification designation.
Antonospora.
I have no
You don't know
idea.
A-N-T-O.
I know, I saw it too. I didn't
No?
recognize it.
And nematocystis, we know. Anyway, there's a table of how these guys,
They're all protozoans.
what they do with respect to heme.
Every
how
single
they
one of
make
these
it.
is listed as a protozoan.
So they're ignoring the nematodes
And the cestodes and,
and the trematodes?
Yeah,
That's unfair, right?
I thought so.
Or unbiased. It's biased because it doesn't give you the whole picture. Why are they ignoring the worms, basically? Why are they doing
Because
that?
they're not trained in that.
Oh, they don't know anything about it?
Well, they might be able to review the literature, though. I would love to see what they had to say about that, because I was looking forward to them explaining the presence of the uptake of vitamin B12 in tapeworms, for instance, which is one of these heme-containing compounds. They sequester
don't you,
all of your dietary vitamin B12.
You should write them and say, what about the worms?
I will.
Why don't we do this?
No.
From
let,
TWiP.
Let them listen.
No, they're not going to listen. There's just no way.
We'll send them a link and say, please listen to our podcast, because in it we thought you should
We
have
could just ask
expanded
them the
this
question
review.
and then read their answer next time.
We could do that.
They go through a bit here where they talk about how these parasites, just the protozoans, right?
Yes. And some free living.
Make their heme. They say apicomplexan parasites have an unusual type of heme synthesis. So the apicomplexans make heme.
Which is interesting, since malaria
Yeah.
tries to get rid of your heme.
But it wants to make its own.
Yeah, that's fascinating.
Weird. And it says it spans 3 cellular compartments, and uses enzymes of various origins, in other words.
It's amazing.
Its own and stuff it's acquired from endosymbionts, right? Like the apicomplexan was an endosymbiont.
Wild.
Right?
Yeah, sure. That's wild stuff, isn't it?
Mitochondrial pathway, cytosolic pathway, etc. And they say, in contrast to the apes, many other groups have lost the capacity to synthesize
So
heme.
they don't have de novo synthesis.
They don't. And they say this makes sense because anaerobes don't need it.
What is that called, Vince?
What?
Salvage pathway. The organisms that can't make heme have a salvage pathway.
They get it from someone else.
They do.
Like their host.
Like their host.
The host that they parasitize.
That's right.
Take advantage of, right?
They give nothing back. They just take everything. It's
Yeah.
all about them.
They say the colonization of anaerobic environments, which makes heme synthesis impossible because you need oxygen
Correct.
to do it,
Correct.
is probably one of the reasons for this loss. Yeah, that's
I would say
sounds reasonable, doesn't
That's
it?
right. QED.
And they also say anaerobes only need a little bit of heme. And so maybe it's easier to get it from their hosts instead of making it. So I would guess that's energetically.
So
making more sense.
Toxoplasma Trichomonas rather. Does Trichomonas? It's a strict anaerobe.
Trichomonas,
Yeah.
does
What's
it
that-
make its
that-
own?
The table has to show something there on Trichomonas.
Yeah, the ones that make their own are the apicomplexans.
Trichomonas is not one of those.
It's not one
a
of those.
flagellated protozoan.
Leishmania
That's a flagellated
makes
protozoan.
its own. Trypanosomes
That's a flagellated.
make their own. Which one did you ask? Toxo?
Trichomonas.
Trichomonas. Here, that's an anaerobe.
It's an anaerobe, right.
And
It probably doesn't.
does not. No,
Right.
It does not.
Right.
Must get it from the host, right?
Yes. And guess what it secretes as a electron acceptor?
What?
It secretes molecular hydrogen.
Oh, yeah. Christian Deduve figured that out, right?
Well, Miklos Mueller.
Miklos Mueller?
Yeah, I was a colleague of his. The point is that Craig Venter has spent his entire fortune almost cruising the oceans looking for organisms that secrete molecular hydrogen. And here's one sitting right here.
Yeah. Trypanosomes take up heme from the host.
Yes.
Leishmania and insect trypanosomatids use later precursors of heme synthesis as they encode enzymes for the last 3 steps of the pathway. So they take not heme, but a late precursor and then...
Protoporphyrin 9
Yeah,
or something.
exactly.
8,
The
7.
genes that do this in Leishmania have been acquired from gamma proteobacteria by lateral gene transfer. Isn't that incredible?
That's amazing. That's amazing.
So
Now-
can you name a famous gamma proteobacteria?
no.
Have you had any hamburger lately?
Oh,
E. coli.
No. Oh no. Oh no.
E.
Why is
coli,
it?
Pseudomonas.
Why is it called that?
Gamma? The bacteria are classified into alpha, beta, and gamma, and delta.
I've missed out on all that as an adult. It just bypassed my knowledge base.
Some insect trypanosomatids harbor bacterial emzosymbionts that supply them with heme or its
Sounds like Wolbachia.
Isn't that incredible? So then this question is, do all parasites require heme?
Aha.
The requirement for heme usually correlates with metabolism, whether or not you need oxygen. So anaerobic species possess only a few hemoproteins, mostly involved in sensing these gases to protect them, as we said earlier.
Right.
And Amoeba has no heme protein, but it does secrete two proteins that binds heme in vitro.
Really?
Scavenge it, maybe.
Huh.
And they think that might be because of the requirement for iron. So heme binds iron. Right.
Well,
so
some do.
Thinking that
Some
maybe
do.
Entamoeba is getting heme to get the iron and not the heme itself. I don't know.
So
about
it can
that.
make ferredoxin and stuff like this.
What is a biotroph? Do you know?
Troph or trough?
Troph. BIOTROPH.
A biotroph.
The aforementioned parasites are biotrophs that cannot be cultivated in chemically defined media, meaning that their dependence on heme cannot be tested. The only known eukaryote able to prosper without heme is the kinetoplastid parasite of plants, Phytomonas.
Okay, I missed that too.
So apparently it's hard to tell if some of these guys need heme or not because you can't grow them.
So it's not even... I misspoke before. I thought the Phytomonas was a free-living parasite, but it's not. Not a free-living parasite, a free-living protozoan. Okay, it's a parasite of plants. As Crithidia are also.
Yeah, the statement free-living parasite is wrong.
Right, you can't be a free-living
Only
parasite.
a headline writer would write that.
By the way, there's a free-living stage of a parasite, though. You can have a free-living stage of a parasite.
Oh, yeah, of course, a stage, but not the whole life cycle.
No.
It's not a parasite
Then
anymore.
it wouldn't be a parasite, then, would it?
What's a parasite of Earth?
We. You and me.
We are taking something from Earth.
What are we giving back?
So the definition of a parasite is one organism that benefits at the expense of another.
Yes, but by internally living...
You could be on the surface, can't you?
Not really.
These ectoparasites that live on your skin, they take something, they benefit from you.
We wouldn't get into a semantic argument on this one.
All right, but Earth is not an organism, is it?
Of course not. Unless you believe in the Gaia hypothesis.
But it's made of bacteria and a biome. I think it's an organism.
You do.
I think the rocks and the sands and the dirt
It's
are
alive.
the superstructure that hold the organism in place.
I
It's
would
like
agree.
a crab. The shell is not living.
I am not
but it
disagreeing
supports,
with you.
Oh, I don't have to go on?
No, you don't have to go on at all. You can stop right there if you'd like.
10 years ago, I would never have voiced this idea.
Right,
But
but-
talking to you for a while has really influenced me.
Well, and vice versa, by the way. In a good way.
Listen to this statement. Maybe you can explain this to me. I highlighted it so that you can...
I've missed several others, but go
All
ahead.
heme functions can potentially be lost or bypassed when so much energy is provided by glycolysis that oxidative phosphorylation-derived ATP is not required. Do you understand that?
Yes.
So if you need oxidative ATP, derived ATP, you need to have heme as a,
Right.
what, an electron acceptor?
Yes, that's right. That's right.
But if you can do it by glycolysis...
Yes. But that's a very wasteful process.
Oh, I'm sorry. Oxidative phos requires oxygen.
Well, of course.
That not terminal acceptor. No. The oxygen is what heme provides for the production of ATP by oxidative phosphorylation.
That's right.
But if you can make a lot of energy by glycolysis, which is
Yes.
anaerobic, right?
Yes.
It's like fermentation.
That's right.
Then you don't need heme. Got it. How do parasites cope with heme toxicity? This is the good part, right?
Yes, it is.
While useful for many cellular processes, free heme induces the production of reactive oxygen species and causes lipid peroxidation, resulting in membrane injury. So a lot of heme is bad. The large quantities of heme released during hemoglobin digestion, who does that,
Malaria parasites.
are a problem.
Yep.
It's a challenge to parasites dwelling or feeding in or feeding on blood.
So they have a special mechanism for stacking up the hemozoan portion of hemoglobin, and interfering with that enzyme results in the death of the parasite.
They call it detoxification. breakdown of heme
That's
into
right.
molecular iron and less
That's
reactive
correct. That is correct.
intermediates, containment of heme by a physical barrier,
Yep.
and the conversion of heme into an inert crystalline structure.
Which is what malaria
That's what
does.
you're talking about.
That's
And
right.
The formation of hemozoan.
That's called malaria pigment. When you look at it under the microscope, it's called malaria pigment.
So that's a single unit heme polymer. So that's a way of detoxifying the heme, right?
Correct. So they will stack up in certain regular orders.
It says that schistosomes also do that.
Haha.
And
They regurgitate it.
They say they make these hemozoans.
Yeah, and then they regurgitate
it. They also say the blood-sucking triatomine bugs do this.
The ones that carry Chagas disease.
And so the bugs actually do this to heme because they don't like it's toxic. Interesting, isn't it?
Yeah, well, too much oxygen. Not good.
The ability to break heme is still questionable for many parasites. It's usually done by heme oxygenase, which leads to the production of biliverdin, which is then metabolized to bilirubin, a non-toxic water-soluble
Do we make bilirubin,
pigment.
Vince?
Man, our livers.
And then what do we do with it?
It goes to the bile ducts.
And then what we do with it?
It goes into our intestine.
And then what we do?
We excrete it.
Yes. What else do we do with it, though? If it becomes soluble in our blood, what do we do with it?
Don't know.
Convert it to urea
And
in the bladder.
then it's excreted.
Yeah.
So this is a metabolite of heme in us?
Bilirubin comes out in your urine, too.
So if you have too much bilirubin, you have colored urine, and that's a sign of what?
I do.
Some liver disease?
It's a sign of liver disease, that's right. And that's why your stool is that color.
Because of bilirubin?
Yeah, and biliverdin. That's right. And that's where urea comes from, too. And that's all about the breakdown of hemoglobin.
Isn't biology amazing?
It's fantastic, because what you're doing is deconstructing David Shemin's synthesis.
Yeah.
That's exactly what you're talking about right now. David Shemin spent his life learning how the protoporphyrin 9 molecule is put together, and here you are telling how the parasites and other processes take that molecule and detoxify it by breaking it down into what it was before that.
It's very cool.
I love it.
By the way, heme oxygenase has recently been identified in Plasmodium, but whether it's in other parasites is not known.
Yeah.
Say Leishmania seems to be able to break down heme to use it for iron. I think we mentioned that before.
We did. In malaria, though, they create a toxic waste dump of heme in this hemozoan. It collects in the middle of the cell, and that's why the parasite doesn't divide until the very last moment. Doesn't that sound weird?
Yeah.
You have a red cell,
Yeah.
you've got 8 nuclei, they're all connected together by the same cytoplasm.
Right.
And in the middle, you're getting this crystal of hemozoan developed, right? And at the very last moment, the parasite culls off 8 different cells, depending on the species, and leaves behind the hemozoan as a toxic waste dump. Then when the cell bursts open and releases the parasites, the hemozoan goes out too. And
What happens to that?
It becomes a toxin. It eventually ends up in the liver.
Yeah.
It's sequestered by the Kupffer cells. And when you do a section of liver of a patient that's got heavy malaria infection, you can identify every Kupffer cell because that's where the hemozoan is. And it's almost an autofluorescence. It gives a very shiny-like appearance under the microscope, so you can spot it in a minute.
And we can't deal with hemozoan.
We cannot. So there's another way to detect extremely heavy malaria infections.
Right.
And that's to look in the neutrophils for hemozoan.
Cool.
When you see that, you whole body transfuse the patient.
So remember, everyone, hemozoan is a way for the parasite to deal with heme, which is in the blood, of course, and it's toxic.
Well, it eats hemoglobin, but it cleaves off the globin portion.
Yeah.
And it leaves a small peptide behind.
And attached to heme.
It's yes.
And
Otherwise,
the heme is toxic,
otherwise you just call it heme. That's why it's called hemozoan rather than
and
heme.
Heme is toxic.
Very, very toxic.
Free heme induces the production of oxidative radicals. Okay.
That's true. So worms produce heme too. Lots
They
of
produce
heme.
their own heme?
They do, like Ascaris, for instance. Ascaris.
Okay, so they have the genes for the whole thing.
They do. And
They don't
Trichinella.
scavenge it from the host as
They
some
do
of these
not.
guys do?
They do not.
No?
The
Okay.
worm that I worked on, Trichinella,
Yeah.
has 2 distinct stages, right, as we reviewed way back when. It has a stage that lives in the muscle, and it has a stage that lives in the gut tract.
Okay.
The adult worm has no heme whatsoever. 0.
The adult worm doesn't make it
I don't know that.
and doesn't acquire it. It just doesn't have it in it.
It's apparently not. They're transparent. They're not colored.
Why is that? It doesn't need to use oxygen?
I don't know. No, it's a microaerophil. In fact, this worm is a microaerophil.
So it doesn't need heme.
So maybe it makes it in its mitochondria, but it doesn't exhibit the color of heme. Whereas the worm that lives in the muscle, the intermediate stage that you swallow with raw meat, if you collect them into larger than, let's say, a quarter of a mill's worth of worms in the bottom of a test tube by digesting the carcass of a mouse, guess what color these parasites are?
What?
They look like salmonella.
Uh,
They're pink. They're pink. And the pink is due to the hemoglobin.
Okay.
And the reason why I know this is because I used to do a lot of electrofocusing, flatbed, preparative isoelectric focusing.
So this is a kind of electrophoresis that you're talking
yes,
about.
in a starch granuled modification, so you can load up a whole bunch of stuff and then slice it up after it electrophoresis, and you can...
What's the purpose of doing this?
Is to isolate the proteins separated from each other in large amounts so you can test out their functions. Well, two of the bands that used to form all the time, every time I would make a cell-free extract of trichinella, was this green band and a red band. I can show you pictures, Vince. You'd be amazed. And they're both hemoglobin-like molecules.
It's a Christmas gel, right?
Just about right. And the purpose of it is to keep the oxygen away from the worm's machinery.
I see.
It's a strict anaerobe. The worm is a strict anaerobe, and it generates a lot of those molecules so it can trap the oxygen so it doesn't hurt the parasite's biochemistry. It's amazing.
Cool. That's very neat.
Yeah. That's why I was disappointed. I didn't see any reference to that here.
We'll send them.
But I think it's because they just covered the protozoans. Maybe there's a second article coming out that will talk about the heme.
The title says parasites, so
I know that's why
you could argue that these are not all parasites. Right?
No, these are all parasites.
I'm sorry, these are not all the parasites.
No, of course not. These are just protozoan parasites.
Yeah, exactly. And they should have put protozoan in the title, right?
If they only talked about them, I think that was probably right.
I wonder who handled this review. Let's see if there's a person here at the end. We can...
Peter Hotez?
No, it's not here. So the last part here is heme metabolism a good drug target?
Right.
And so they say that it's been proposed for Plasmodium, which encode their own heme synthesizing machinery. But they say that if you inhibit that, it's going to get it from the host. So it's probably not a good... Yeah, I would say maybe. Who knows, right?
It takes host hemozoan and stacks it up and gets rid of it.
Because in order to get it, you have to be able to import it.
Right.
Which requires transporters.
And
right?
Just for that molecule. And that's not going to happen with Plasmodium. I think they're wrong in that.
Yeah. So
David Fiddick can help us out on that one.
Because they argue that in say, the anaerobic cryptosporidium, it's lost the capacity to synthesize heme, but there's an organelle that wait, hold on. The anaerobic crypto has lost the capacity to synthesize heme, and the apicoplast, which does it, is gone. So in Plasmodium, the apicoplast is present because you need to synthesize heme. They say, nevertheless, we argue that Plasmodium heme synthesis is not a suitable drug target because you could get it from the host. On the other hand, the pathway of heme detoxification is a good target for malaria.
Yeah, well, that's
quinolin, apparently.
Yeah, chloroquine
inhibits hemozoan formation. Did you know that?
I did.
I did not. It's also effective against schistosoma.
I knew it.
Which make hemozoans
but the protozoans are resistant to that drug. Too bad. That's chloroquine and mefloquine and all those quinolins derived from quinine. That's how it works.
They also say that Leishmania, remember, encodes the last 3 steps of heme synthesis. Maybe those enzymes would be good targets.
Yeah.
Importers, the parasites that import heme from the host, they say you need specialized transporters for this, and these may be good drug targets as well.
You bet.
So the pathway of uptake. So there you go.
Yep.
Well, I thought this was nice because it sort of
gives us
an overview of heme. But as you say, it doesn't do the worms. And I think you ought to write the article, Dickson. You have no interest in doing that, right?
I don't know. I'm not a knowledgeable person in that field.
Hold yourself up in your man cave for a few weeks. Read all the literature.
You're right.
And then write it's not worth it.
Why not? No, I didn't say it wasn't worth it. I think people need to know. What you did by picking this article, though, was it allowed a wider discussion to occur than just about the protozoans. So that's good.
Okay, let's read a couple of emails here.
Sure.
The first one is from Jim. who writes, Professors, you've discussed this idea before, but I thought you'd enjoy this nice summary from Nature. It is a summary called Autoimmunity, the Worm Returns.
Aha.
Joel Weinstock explains why several clinical trials are deliberately infecting people with helminths to treat autoimmune diseases.
Right.
We have talked about this, right?
We have.
These are great immunosuppressive guys, right?
Or they immunodivert. Here's what they do. They redirect the immune response, which was originally selected for attack against the worms. If you take the worms away, it finds other targets that are similar, and that turns out to be you. So what the worms do is they return the immune system to its original target.
Mm-hmm.
And that prevents host damage, of course, because now the immune system is totally in tune with the antigens that they're
So the example
for.
in this article is inflammatory bowel disease.
Yeah,
which is being treated by giving worms. Right?
And successfully.
And I do enjoy your podcast. Thank you for sharing your wonderful conversation. Sincerely, another Jim from Virginia. So TWiV and TWiP often gets email from Jim from Virginia.
Really?
And this is another Jim from Virginia. How about that?
That's amazing. There must be lots of Jims in Virginia. A lot of people working out over there.
The next one is from Alan, who sent a link to an article in Scientific American Blogs. Let's see who it's by. Catherine Harmon. Parasitic worm eggs ease intestinal ills by changing gut macrobiota.
Right.
Wow.
Yeah, I've read that one.
This is also about inflammatory bowel disease.
Right. You remember we discussed trichuris attaching to the gut wall by a bacterial bridge.
Trichuris trichiura.
Yeah.
So they have a monkey model for
Right.
IBS, and they give them trichuris, and they improve.
Yep.
And it's the same idea? Immunosuppressive?
Same idea. Not immunosuppressive.
Immunodivertive.
Immunodivertive.
It says here
exactly
that
right.
the trichiura restore the balance to the microbial communities that are attached to the intestinal wall.
Well, that may also be true, but here's what I really think is going on, okay? That's my own opinion here. I'm not disagreeing with this article at all, but I'm saying that once the immune system stops attacking the host, the gut tract has the ability to return to normal, which would, of course, include normal floral habitat
Could be that
restoration.
That's the mechanism, right?
So it's habitat restoration is what you're looking at here.
Our last paragraph is, so if parasitic egg treatments prove successful and do eventually make it to market, don't be too squeamish. Perhaps just think of them as the caviar of probiotics. Thank you for that, Alan. Virginia writes, I've been fascinated with parasites for years, ever since reading Peeps, a novel by Scott Westerfield. While it is fiction, every other chapter focused on a particular parasite and described its life cycle. You know this book?
I don't.
When I found this podcast, I nearly burst from happiness. While I'm no longer a wildlife biology student, I recently switched to an English major. What a change, eh?
I'll say.
I still love learning more about parasitism. So many people find it odd that I'm so enamored with such gross creatures, but I think they're just nifty.
They are.
The caviar. Anyway, I just wanted to thank you for starting this podcast. It is both informative and entertaining. Keep up the excellent work.
Thanks. We will.
Ethan writes, saw this, and it instantly reminded me of Dickson's heavy breathing in the background.
Oh, no.
He sent a poster that reads, getting angry at people when we listen to them eating or breathing is called misophonia, which is actually a brain disorder.
Oh, good.
So I have misophonia.
Who got mad at me?
Me. It's me all the time. I'm always telling you to stop breathing. Misophonia, literally hatred of sound, is a form of decreased sound intolerance. It is believed to be a neurological disorder characterized by negative experiences resulting only from specific sounds.
Goodness.
I don't know why I would have negative experiences against breathing. I just want the audio quality to be pristine.
This is true.
Don writes, Dear Sirs, I became interested in parasites of the gills in freshwater fish, especially Myxoma fundibly, and was surprised to learn that this animal produces spores. How common is that? Your TWiP MV is marvelous. Thank you so much. All right, let's look this up.
Myxoma. Is that a microsporidium?
No, there's nothing that matches.
Myxoma.
Nope. Myxoma Fundably is not is not a word. It's not picks up.
What about
myxoma is a kind of tumor, of course.
Right. I've never heard of this one.
So I don't know if he means that the parasites of the gills in freshwater fish form spores or what. Do you know of parasites in the gills of freshwater fish?
I do. I know a lot of them. But
What are they
they're all
called?
monogenean trematodes.
Do they form spores?
No, they don't.
So we can't help Don.
Not at that level.
Don, give us a link.
If it's a microsporidium, right, and there are lots of them, and they're entocytozoans,
Yeah.
encephalocytozoans, sorry, then those organisms are sort of a bridge between bacteria and protozoans. They're an intermediate group, and they have weird life cycles. And I knew someone who actually studied them. They're very important for certain marine organisms in terms of the economics of, like crabs, for instance, and oysters and that sort of thing. And they produce weird-looking spores, right? Nematocysts, and then they almost look like a nematocyst that you'd find on a coelenterate, like a hydra. And there's a little coil inside that looks like a flagellum, and in order to hatch, it's like a spring. And then you get fired out of these little cysts, and that's how they infect the next host. But if this is a microsporidium, then it's totally explainable by the fact that they do form spores.
Here's a disease, neon tetra disease, is caused by a sporozoan.
What's a neon tetra? Do you know what a neon tetra
It's a
is?
fish.
Of course. Where does it come from?
I don't know, but I used to keep them in my tank.
Me too. So did I. You know where they come from?
No.
They come from the Amazon.
Is that right?
They're freshwater fish.
No, I didn't know that.
And
That's what he's talking about. This disease is caused by a sporozoan, which is a parasitic spore-forming protozoan.
okay.
And they reproduce by means of spores. So is that unusual to have a spore-forming
Well, you'd
protozoan?
call it a spore, but it's more complicated than a bacterial spore. So I think it's probably a cyst-like production. But it's a single cell, so you can't call
The
it a cyst.
spores enter the tract, and the newly hatched embryos burrow through the wall. So not really spores, are they?
No. Neon tetras, though, I want to come back to that because it was an interesting story about how they're collected. When it rains in the Amazon for months and floods back into the forest, the Amazon River, in the middle of the Amazon River, remains muddy. But because the water settles out in the periphery as it floods through the forest, the water forest, as they call it, or the tropical water, I think that's their term. These tropical fish invade that region, and now they're in crystalline clear water. They're absolutely beautiful. And neon tetras come from there, and so do all the other tropical freshwater fish that you can buy, except the cichlids, which come from Africa.
Right.
So they showed a person who was commercially selling these fish, right? So they would collect, let's say they collected 1 million of these fish,
Right.
With big drop nets that they would let them swim underneath and then lift them up and put them into small bags, and then they would find this mothership out in the Amazon River waiting for this shipment, and then they would take them and ship them to the port of Belém, and then Belém would fly them to Los Angeles, and they would sell them in the stores. You know what the survival rate is? It's about 1 in 1,000.
Wow.
And the price of this fish just
Terrible.
kept quadrupling as it moved up the economic food chain, basically. The guy who caught them got almost no money whatsoever. And the end result, of course, the pet shop gets about $1 a piece or something crazy like this. And if you looked at how much this guy got for selling his 1 million neon tetras to the mothership, it was like $1 for the whole shipment. Excuse
Terrible.
me. Yeah, it is awful, because if you look at how much they're really worth.
Yeah. Do they still import them, or do they breed them?
That's a great question. I think I'm under the impression that they're still imported, but I don't know the answer to that one. Other tropical fish collectors would know.
You gonna cough? All right. Just don't cough in my face. I'll have to edit that out.
That's okay. You can leave it in if you'd like.
Spencer writes, Dear Vincent and Dickson, here's a link to an interesting study published in the Journal of Clinical Endocrinology and Metabolism that was done in China, suggesting that schistosome infection may curb diabetes risk. The researchers detected an association between previous schistosome infection and a lower prevalence of diabetes in patients. This may hint at a link between glucose metabolism and T-cell mediated cytokine secretion. Before I tell patients to go seek out schistosome infection, perhaps more studies need to be done. Oh, so the title is Association of Previous Schistosome Infection with Diabetes and Metabolic Syndrome, a study
This is
done
type 1 diabetes?
in rural China.
Type 1 or type 2? Diabetes.
I have to look at the paper. I don't know.
Aha. Because, I mean, you can imagine how, again, immunodiversion, if the schistosome antigens, and there are lots of them, if one of them resembles the same epitopic protein that the virus, we think, kills off the beta cells from the islet of Langerhans, then schistosomes would act as a diversion away from the islet cells and keep the immune system busy trying to attack a parasite rather than the islet
I
cell.
believe it's type 2.
Type 2, that's an adult. Different, totally different. I take it all back.
Metabolic syndrome.
No, take it all back.
Yeah, combination of disorders, type 2.
Right.
So there could be other confounding factors.
Well,
that
his glucose explanation might be right then.
do this. So it's interesting, though. But obviously, when you give people parasites for therapy, you have to be careful because we don't know everything.
I wouldn't give schistosomes for any therapy. It's an awful
But
disease.
hookworms are okay.
Hookworms are okay because there's nothing permanent about the hookworm infection.
Unless you're immunocompromised?
No, even if you're immunocompromised, there's nothing permanent. Whereas schistosome eggs can lodge in your brain and your spinal cord. It could be awful.
All right. So it's a bad idea.
It's a bad idea.
Schistosomes. All right. Newer writes, I apologize for the large volume of correspondence and for not knowing the entire catalog of what has been and has not been covered. I send these emails with only the utmost respect and admiration. So he's referring to the Candiru myth.
The Candiru, okay.
You know what that is?
The little parasitic catfish.
Yeah, that swim into your ureter.
That's right.
Of male or female.
Yeah, both.
And he said, I was so ready to roll my eyes back into my head for that story, but found myself captivated. I believe I sensed the gift. That's your gift of storytelling, Dickson. Any chance of exploring the facts? So he sends an extract from Wikipedia, which is about William S. Burroughs, who wrote about the Candiru in Naked Lunch, right? 1959 novel.
Ugh.
A small eel-like fish or worm about 1/4 inch through 2 inches long, patronizing certain rivers of ill repute in the greater Amazon basin that will swim into your ureter.
It's not incorrect, but it's not an eel-like thing. It's a little parasitic catfish. It's a tiny, tiny, tiny little parasitic catfish. And what it's really looking for are the gills of giant catfish.
It's not a parasite, or it is a parasite?
It's an ecto... you wouldn't call it a parasite in that sense. You see, that's what I'm saying. It any more than you'd call a tick a parasite, or a louse a parasite, or a mosquito a parasite. They come in, do their job, and leave.
Mm-hmm.
So what would you call that?
Do they...
There's no permanent association in the gills
No,
of the...
but do they take something from the host without giving back?
Of course they do. So
then
that's
a parasite, right?
Well, okay.
So he asks, Newer asks, not knowing anything about its reproductive or life cycle, it's difficult to say if this should be classed a predator or parasite.
Right, exactly.
But in my book, anything that allegedly swims up your urethra and needles itself in there permanently should be classified a parasite.
No, it's just
one that certainly shows no regard for the life of the host.
It's an aberrant localization.
Yeah. It's an
of
accident.
A parasite.
Well, so is toxo an accident, right? Infecting people.
No, but that's different. This little parasite seeks out urea, the breakdown product of hemoglobin, which gets us back to David Sherman. No, that's just a joke. So this little catfish doesn't stick around, though. It actually takes all it needs and then leaves, and then when it needs some more, it goes finds another catfish. And the reason why I can find those catfish, I mean, some of these catfish they have got a head this big, Vince. I'm almost making my hands touch. All right, I'm making a big circle
with your arms,
I am
touching your tips of your fingers.
Yes, I am,
right?
And that's how big around the head of some of these Amazon catfish are, and they live in water that you can't see in.
Yeah, it's brackish.
So
it's
dark
brackish,
water.
Dark. Yeah,
dark water. So fish in those zones find each other by electrical signals, basically. Mm-hmm. Okay. This little parasitic catfish, though, they call it a parasitic catfish. It finds its host by following the trail of urea until it gets concentrated.
So that's why it goes into your urethra. How often does this happen, Dickson?
It doesn't happen often with tourists because they're warned about this when they go down there. When you go swimming, make sure that you wear underwear under your swimming trunks. And never, never, never urinate in the water.
Okay.
Like little kids in the swimming pools or in the bathtubs. Don't do it. Because if you do that, you'll set up a trail of urea.
It doesn't feed on the membranes, does it?
No.
Because isn't this
it rasps.
Burroughs? It says it feeds on the soft membranes. No,
it doesn't. It dies. It actually goes into the ureter and dies. It doesn't.
And then
live.
you have to have it removed?
You have to have it removed.
Surgically?
Yeah. Because it's got spines. It's got these little fins.
Yeah.
Okay. I can't articulate the face. I can do it like this. They only bend down when the catfish is going forwards, and then when they try to go backwards, they're like little barbs.
Sure. Yeah.
And they hook and they stick.
Does it obstruct?
Yes.
So you can't urinate?
I'm not sure about that, but there's almost no room for the urine to go out.
Oh, well, you need immediate help if this happens.
You do.
Because within a few hours, you're going to have a full bladder and
This
you
is
can
all
rupture,
true.
right?
This is all true.
Wow.
It's a bad thing. You can look it up. I mean, they've got a whole literature on this, but
he says that it was a major plot point in an episode of the animated adult swim show, The Venture Brothers. I've never heard of that.
I haven't either.
But can you imagine this being a plot point? I would guess so.
Well,
it's very
have you ever seen a movie called The Medicine Man?
No.
With Sean Connery?
I have not.
There was a movie he made I didn't think it was one of his best movies, about the Amazon jungle and drug discovery. Okay, and a pushy New York female scientist that goes down to find out what this guy is getting out of the jungle that they're hiring him to get, that cures a certain cancer, let's say. I forget the actual details of the plot, but when they go down there, of course, there has to be a romantic interlude, right? Otherwise, it wouldn't be a Hollywood movie.
Mm-hmm.
And in it, there's a scene where they go swimming. And Sean Connery is quoted as saying something like, "Make sure you keep your knickers on to avoid Kanduru." I can't say it like him, but I can.
Kanduru. Is that right? Kanduru.
Kanduru. Canduru, I believe.
He spells it with an I.
Okay, doesn't matter. If you look it up, you'll find
All
a
right.
whole literature on it.
Next one's from Robin. It's our last email.
Yeah.
With regard to TWiP, TWIM, and TWiV, keep them coming. Good. Possible reservoirs in a vertical farmer. So a long time ago, we talked about how the people who work in a vertical farm might contribute to infecting.
Go on.
Right? Hair,
Yeah.
feet,
Yeah.
toes,
Sure.
bottom of your feet,
Yep.
your legs,
That's right.
pubic hair, ducts of all sort.
Unusual, but...
So we carry a lot of organisms.
Oh, but of course
obviously.
we do. And the way to avoid that is to change into a barrier suit.
Yeah, you wear a barrier suit. So is that what you'll have to wear in a vertical farm? A Tyvek barrier suit with a powered respirator?
No, not a powered respirator. In Japan, for instance, they have what they call plant factories now. And plant factories are more than one story. So those are classified as vertical farms. And if you go to any of the websites for like New Veggie, and there are a couple of others now out, you'll see all the workers are completely gowned. They wear gloves. They have masks. They have surgical hats. They're totally barrier protected, so they don't want to transmit anything to the plant. The plants are not sterile, but they're clean. Yeah, I agree.
Okay.
I think you have to be careful. Otherwise, you're going to lose your crops.
He had a few other, I think these are miscellaneous things we talked about. Visceral pain. All right, we talked about visceral pain at some point. I don't remember why, but he says all viscera except the central nervous system have pain receptors. They are activated by stimuli appropriate to the organ, which are not necessarily the same as the stimuli for somatic receptors. The intestine can be cut with a knife without pain, but just about everyone has experienced the pain from stretching of the gut. The pain receptors in most solid organs are located in their fibrous capsules and are stimulated by acute stretching or acute inflammation. Robin is a retired emergency room physician.
Great. Thank you, Robin.
Insect, bugs, and arachnids.
Yes.
It was quite clear to me in school that all bugs were insects, but that not all insects were bugs. I would not have graduated if I were not clear on that point.
Let's just put it this way. All insects are arthropods. Okay, but there are lots of different arthropods out there, right? There's crustacea, there's a whole bunch of things. So arthropod means jointed legs.
We talked about this.
Yeah, let's start with the phylum Arthropoda, or Arthropoda. Mm-hmm. And then underneath that, you've got all these categories of things like crustacea, etc. You've got Arachnia, which are the spiders, right? You've got ticks and mites, right? And then you've got the insects.
Right.
Now, under the insects, you've got orders of insects. Let's take common ones that everybody knows about. One is called Diptera. Okay, those are the houseflies.
Right.
But they're also the mosquitoes. Mosquitoes only have 2 wings. So if they have Diptera, Diaptera, that's the way that goes. Okay, so you've got the Diptera. Then you've got the beetles. Those are the Coleoptera. Okay? And then you have bugs, true bugs. And they're called Hemiptera. You referred to one before, the Triatomids. They have big siphon-like biting mouthparts. All of them have this. Every one of those bugs has that as their means of obtaining their food. And there's a lot of others, too. There's Columbella, there's Orthoptera, there's Ephemeroptera, which are the mayflies, and the Trichoptera, which are the caddisflies, and the Plecoptera, which are the stoneflies. I had to know all of these at once because I took a course and I got my degree in biology.
And you remember this, though.
I do know that Siphonoptera are the fleas.
So people use bugs in a colloquial way.
They get the bugs out of it.
They even call them bacteria bugs, right?
Tell me about it.
It's terrible.
There was a course here given by Harold New and Paul Elner. Paul Elner was in charge of the diagnostic microbiology lab, and Harold New was an infectious disease physician. And guess what the name of their course was that they gave together?
What?
Bugs and Drugs.
That's good. I like
that.
But
But bugs, in this case, they were referred to.
They shouldn't use bugs.
Well, bacteriologists use it all the time.
I don't like it.
I don't either because obviously it's incorrect.
A bug is a certain
It's
thing.
imprecise.
He continues, I had to unlearn English after coming to the U.S. of A. He writes the U small letter.
Yes.
Lowercase u, as that is the way it was originally intended in the Declaration of Independence. Likewise, I would not have graduated if I had lumped ticks and mites with insects. But of course, in the U.S., one must not expect to hear about medical arachnidology. Mosquito swarms. In my childhood, I remember swarms of male mosquitoes swirling above the heads of people in the evenings. One knew they were male because they had mustaches, feathery antennae. Therefore, one was not concerned about being bitten by them. Is that right?
It is correct.
Male mosquitoes have feathery antennae?
They have larger, brushy antennae than the females.
How could you see that in a swarm?
I don't know that one, actually. You'd have to ask him.
Well, I believe him. It's okay.
Well, it's pretty good eyesight to see that one.
This episode of TWiP will be found at iTunes, as usual, and at microbeworld.org/TWiP. And if you do like TWiP, please go over to iTunes and subscribe if you haven't, and leave a comment or just rate us over there. That keeps us prominent so we can get more listeners to hear the fable storytelling of Dickson. Could you tell a story sometime that we haven't heard on TWiP? Do you remember once you read one of your stories?
I did. I haven't written too many stories lately.
You can't tell any stories.
I could tell stories. I can tell some stories.
People like to hear you tell stories. Maybe next time you could do that.
I can think of one. I'll try.
I'll try to be nice. It'll be a nice story.
It'll be nice. It'll be nice.
Send your questions and comments to twip@twiv.tv. Dickson Despommier can be found at trichonella.org, medicalecology.com, and verticalfarm.com. Thank you, Dickson.
You're welcome, Vince.
Good to have you back. Hopefully we can get a bunch of these out.
I feel like I'm back in the saddle.
You're always welcome here at TWiP.
Oh, I should hope so.
I'm Vincent Racaniello, and you can find me at virology.ws. Earlier in the show, we made veiled references to a headline writing fiasco. Today, I just noticed a few headlines in the Times and another newspaper that were wrong, having to do with H5N1 influenza, and you can find that at virology.ws. By the way, the music that you hear at the beginning and the end of TWiP is composed and performed by Ronald Jenkins, and you can find his work at ronaldjenkes.com, RONALDJENKEES.com. All his work is there, and we use his work with permission, actually. I asked him a long time ago if we could use his music on TWiP. And it, of course, was brought to my attention by my son. You've been listening to This Week in Parasitism. Thanks for joining us, We'll be back soon. Another TWiP is parasitic.